Good day, viewers. This is 300 Plus Academy, where all we do is exams tutoring, ensuring that you ace your next exam. Still on the business of WAEC, this time around, we'll be talking about biology, practical, taking a look at a final revision. But before we get into things, appreciation. To all of our subscribers, to all of our viewers, for all of the comments, for the words of encouragement. We appreciate that, for always pouring us on, encouraging us to get better and do more. We appreciate all of that, not taken for granted. Still on the business of WAEC 2024, what are our specimens and what are the probable questions? That's going to be the approach. Let's get right into it. As you can see on your screen are a list of specimens from A to Q. Now, looking at a list of these specimens and, trying to, come out with probable questions for this exam, we've been able to group them into four. A to E has been put in a group that talks about various internal organs of various animals. If you look at specimen B, that talks about the vegetative part, talking about vegetative propagation, various modified stems, and all of that storage organs. That is for group B. For group C, that talks about some bones, talking about the thoracic vertebra, the humerus bone, the femur, that's the thigh, and then the lumbar vertebra, that's another group. And then the last group talks about specimen N and R, talking about mango seed and orange seeds. So what are the questions going to be like? That is what we have to profile answers to. So moving on into the first batch, that's the first group, talking about specimen A to E. The first questions we are projecting states that, you're going to just have those specimens, some on a petri dish, all spread out in the laboratory. So the first question says, name the specimens labeled A to E. So on the day of your exam, when you talk about Specimen A talks about the gills of a fish. Specimen B talks about liver with the gallbladder containing a bile intact. Specimen C talks about the lungs of a fowl. Specimen D talks about gizzard of a fowl. Specimen E, intestine of a fowl. So for question one, that is just for us to name the specimens labeled A to E. Now, question 2 says, give one function of each of the specimens A, B, C, D, E. So those specimens we've labeled in question 1, we were told to give their function. So talking about gills of a fish, of course, primarily the function of the gills of a fish is for respiration. Once the floor of the mouth of the fish is lowered, water containing dissolved oxygen moves into the mouth of the fish and then to the gills, and the water of course flows out through the upper column, but the gills there is then going to absorb the dissolved oxygen in that water. I'm talking about the gills, as you can see in the diagram on your screen. On the inner curve of the gill arc, you see gill rakers. That gill rakers is used for feeding. So when we say specimen A for question 2, talking about the functions of specimen A is primarily for respiration, and of course it is used for feeding, talking about the gill rakers. Then talking about specimen B is the liver with the gallbladder of a fowl still intact. So the liver is there for detoxification, it is there for emulsification of fat, more of digestion of fat. It is there for manufacture of bile, where various chemicals in the body is converted into bile salts, and of course for deamination of protein. Usually in animals there are usually no protein stored because of course amino acids builds up protein. So excess amino acids that can lower the pH of the system. So when we have excess amino acids in the system, when blood flows to the liver, those excess amino acids are being destroyed and converted into nitrogenous waste. In the case of mammals, we talk of conversion of amino acids into urea. In the case of birds, like we have for a fowl here, excess amino acids of protein will be converted into uric acid. And of course, in aquatic organisms, excess amino acids of protein via the liver will be converted into ammonia. So these are nitrogenous waste. So what helps in doing that is the liver. So the liver is primarily for detoxification, digestion or emulsification of fat, manufacture of bile, and of course the amination of protein. When we look at C here talks about the lungs of a fowl. The lungs is primarily for respiration, and then D talking about the gizzard of a fowl. As you can see on your screen, the gizzard being muscular, the function is that it helps to grind large food substances into smaller ones, which then move into the intestine. So that is what the gizzard is for. And talking about the intestine of a fowl, that is where the final digestion will take place, having been since the large food particles had already been broken down into tiny particles in the gizzard. In the less muscular intestine, digestion will be completed, and then useful and soluble materials will be absorbed into the body fluid via capillaries. That's talking about intestine. It is for digestion and absorption of nutrients. So that ends question 2. On question 3, as you can see on your screen, it says name the organisms from which specimen A, B, C, D, E are obtained. So where can we get specimen A, B, C, D, and E? That's as they are being laid out on the day of your exam. Imagine being asked in what organisms can you get each of specimen A, B, C, D, E from. When you talk about gills of a fish, of course you can get that from just any fish: tilapia, catfish, titus fish. When you talk from specimen B, C, D, E, these are specimens that you can get from a domestic fowl, and that is why you have that. Okay, for specimen B, C, D, E, you can get all of those from a fowl. And that ends question 3. Okay, moving on to the next question. As you can see, it says we should state 3 observable features that make specimen C so adapted or suited for its function. And specimen C here talks about the lung. So when we are told to state observable features that adapt specimen C to its function, that is telling us to list characteristics of respiratory surface. Okay, and what are the characteristics here? Respiratory surface must be moist. In this case, as you can see, that specimen C, it is moist. Why is it always moist? It is for diffusion of the dissolved gases. Number 2, specimen C is able to perform its function because it is thin walled or thin membrane for easy diffusion. You see, when you touch it, you see that it's always very fluffy, with enough space on the inside, well ventilated. So here it is thin wall, or you say thin membrane for easy diffusion of gases, talking about oxygen and carbon dioxide here. Also, it has a large surface area. Remember, the reason why the lung is there is simply because the organism has a small surface area compared to volume. So this is a problem for most complex organisms. So why we now have the lungs there is to take care of this problem, and that's why it's now conferring on the organism here large surface area. That will increase diffusion rate. And then the other thing you're going to notice that when you touch it, you're going to see blood stain. That is to tell you that, talking about specimen C, the lungs, why it is so well suited or adapted for its function is that it is vascularized. It has an excellent blood supply, so that oxygen that gets to the lungs finds it easy to dissolve in the blood, and of course that way oxygen gets into the blood circulation. So respiratory surface always have excellent blood supply. So these are about, we were told to list 3, so I've listed 4. So then you have to do the needful once you see the question on the day of your exam. Okay, to the next question, talking about question 5 and 6, we can take that together. We were told to state similarities that we can observe between specimen A and C, and for question 6 we were told to state 3 structural differences that we can see between specimen A and C. So this here is talking about specimen A and specimen C. What are the observable similarities we can see, and what are the observable structural differences that we can see? Okay, as you can see the answer on your screen, when you talk about both, gills of a fish is for respiration, that's specimen A. The lungs is for respiration. They are both vascularized, that's they have a network of capillaries, or you say they are highly vascularized. Both are red in color. They are red in color, or you say pink in color. That's because, of course, the blood supply. So when you touch both, you're going to see red blood stains. So that's another observable similar. Both are moist, both are wet. That's another observable similarities. And both are thin walled. When you look at the lungs, just a layer of the filament, you see that it is thin. When you look at the lungs as well, on the inside, there are spaces on the inside. The walls are thin. The walls of the lungs are thin. So both are thin walled, or you say thin membrane. And of course, both also have large surface area. Talking about respiratory surfaces, both are respiratory surfaces. So those are just about the observable similarities. You talk about network of capillaries, or the fact that they are highly vascularized. Both are pink or red in color. Both have moist surface, and both have a large surface area. Of course, for question 6, we were told to look out for structural differences that we can see. Where specimen A talks about the gill of a fish, talks about the gills of a fish. Specimen C talks about lung. Specimen C talks about lung. So what do we notice here? Very easy. For specimen A, gill rakers are present. That gill rakers is used for feeding. For specimen C, talking about the lung, gill rakers are absent. Gill arc or bar are present in A, they are absent in C. Gill lamella present in A, and then are absent in C. Gill filament present in A, then they are absent in C. When you look at specimen A, they are not fluffy, just like a foam, fluffy. But when you look at specimen C, they are fluffy or soft. Yes, they are fluffy or soft. Also, when you look at specimen A, pleural membrane absent. But when you look at specimen C, pleural membrane and cavity absent. So I've listed about 7 observable differences there, and that just about ends question 6. So moving on to question 7, we were told to make a drawing 6 centimeter to 8 centimeter long of specimen A and label fully. As a biology student, you should have a well sharpened pencil and ready to sketch. So quickly, let's get to do that. So let's say I am using my ruler and I'm measuring that this point here is 0 centimeter. This is my 0 centimeter mark, and up to this place, this is my 6 centimeter mark, and then up to this place, this is my 8 centimeter mark. What it means is that whatever it is I am drawing, should not be less than this 6 centimeter and should not be more than 8 centimeters. So all I need to do is first of all start with my gill arc in form of a C, and with the gill arc, I'll ensure that okay, it is not going out of. So once I've done that, then I'm going to just very quickly double that. You can see no broken line. You have to avoid that. So once you've drawn the gill arc, the next thing is that you want to try and draw the filament. And to draw the filament, the best approach is to sort of go this way and then draw another curve on the outside, which of course we are still going to clean at the end of things. So once you've drawn this is going to represent your gill arc. Of course, you then try to get the middle. This talks about the filament. You just go that way. Make sure it doesn't go out of the line. Of course, it's a lot more easier drawing with pencil. So immediately you've done that, the next thing is to take off that line on the outside. It has served its use, and then so that just rhymes. So I've been able to do that. Then the next thing, that's talking about the gill filament. The next thing is the gill rakers on the inside, where I'm just going to draw something like this. I'm going to draw my gill rakers on the inside, some thick, short structures. And that's just about the end of that. Next is for us to label this. Of course, this here is going to be our gill filaments. Now the space in between two filaments is what we call gill lamella, and of course this here is the gill arc, and here that is the gill rakers for feeding. And that's just about the 4 major parts. In the title you're going to write drawing of specimen A. That is talking about the gill of a fish, and that just about ends that. So to the next question 8 says, name the location of each of the specimens B and C in the body of a mammal. Like, where can we find, what's the location where we can find specimen B and C in the body of a mammal? Let's take humans as example. So where can we find specimen B and specimen C in our body? Now, talking about the liver is just after the diaphragm. If this is the diaphragm, and of course this would be the ribs. So if this is the diaphragm, the liver is just somewhere around here. It's going to be this hiatus here, and this here is going to form the stomach. So the liver is somewhere around there. The liver is somewhere around here. This is the stomach. So this muscle here is the diaphragm, as you can see on your screen. So where can we find specimen B in the body of a mammal? Just below the diaphragm. That's upper part of the abdominal cavity. Of course, the abdominal cavity starts immediately after the diaphragm. So we say upper part of the abdominal cavity. Talking about the lungs, of course, we find the lungs above the diaphragm, occupying the chest area. That's the thoracic cavity or the thorax, or you just call it the chest. That is where you can find specimen C. And that ends question 8. So moving on, let's quickly combine question 9 and 10. As you can see on your screen, question 9 says, name the fluid in the gallbladder on specimen B. Specimen B, of course, is the liver. So we're told to name the fluid in the gallbladder. So the fluid in the gallbladder is actually the bile. Yes, bile. And in question 10, we were told to state two functions of the fluid in 9. We are told to state two functions of the bile. Okay, the very first thing you need to know about the bile is that it is an alkaline greenish fluid. It has a pH of about between 7 to 8, around 7.4 thereabout, and it is secreted by the hepatic cells of the liver and stored in the gallbladder. It is usually passed to the duodenum of the intestine. As you can see in that diagram, there is a bile duct that transfers bile into the duodenum. That's talking about the first part of the intestine. Bile has no digestive enzyme but performs, various functions. So for question 10, we were told to list to give at least two functions of the bile. As you can see on your screen, what the bile does is that it emulsifies fats and oils, causing them to break into tiny droplets. Emulsification of fats and oils into fat droplets. Number 2, you can see it neutralizes the acid chyme. The acid from the gastric juice that we have in the stomach. Remember that the gastric juice contains pepsin, HCl, dilute HCl, and water. So one of the things that the bile does is that it neutralizes the effect of the acid chyme coming from the stomach. That's the acid in the chyme is being neutralized by the bile pigment. So that ends that. Also, it provides the alkaline medium for pancreatic juice to act. Yes, that's another one. It provides the alkaline medium needed for pancreatic juice to act. If you see that diagram, you can see where the pancreas is. So the fact that the bile is going to come into the duodenum, the alkaline pH that the secretions of the pancreas needs to act is possible because of the bile pigment that comes to the duodenum. Also, it prevents decay of food in the small intestine. Yes, it prevents putrefaction of food in the small intestine. These are some of the functions of the bile that we have listed. Okay, let's combine question 11 and 12 now. For question 11, we were told to state differences that we can observe between specimen B and specimen C. Okay, between specimen B and specimen C, what are the differences we can observe? That's more like telling us to differentiate what we observe about state observable differences between the liver and the lungs. Now, talking about the liver is a bit more packed, turgid, solid, and thick. But when you talk about the lungs, even when you press it, it's foamy, soft, of we say fluffy or spongy. And talking about the liver, it is darker. It's dark red in color. For the lungs, it is brighter and pinkish in color. You talk about the liver, the surface is smooth all through. The lungs is not exactly rough, but it is folded and a bit convoluted, but not exactly rough. Those are at least the observable differences. Emphasis on the word observable. That's based on what you can observe about those two specimens. And that ends question 11. Question 12. It says make a drawing 8 centimeter to 10 centimeter long of specimen B and label fully. Well, for question 12, do I believe in this question? No, I do not believe in this question, but just for the sake of completion, You can see on your screen the diagram of a liver with the gallbladder intact. But I personally do not believe you will be told to draw question specimen B. I do not believe that, but for those who want to practice that drawing, you have it on your screen talking about the gallbladder, the right lobe, the median lobe, the left lobe. Not much to label, but should you want to draw it, then you can just quickly, practice that. Okay, question 13 says we should state three major vessels serving specimen B. That's three major vessels that supplies blood to specimen B, that supplies blood to the liver. The first one is the hepatic artery that brings in oxygenated blood from the dorsal aorta. That's the great artery. And then you have a hepatic vein that takes out the oxygenated blood, that's blood containing carbon dioxide. And then you have the hepatic portal vein, which is very rich in glucose, that links the liver to the gut, that's to the intestine, so that it is very easy to convey glucose to the liver for conversion of glucose to glycogen for storage. So these are just about the three vessels that supplies the liver. Hepatic artery brings in oxygenated blood. Hepatic vein takes out the oxygenated blood or blood containing carbon dioxide, and hepatic portal vein linking the liver with the gut. And that just about ends that. So to the next question, question 14, we were told in this question to name two diseases that can affect specimen B and C. Still on specimen B and C, we were told to list two diseases that can affect them. Talking about specimen B, that's the liver. We have diseases like infectious hepatitis, liver cirrhosis, cancer of the liver, that's uncontrolled cell proliferation of the liver. Then we have gallstone, a situation whereby the bile pigment, the bile salts in the gallbladder is obstructed, and is finding it difficult to get into the duodenum. It could lead to jaundice and a host of other issues. So these are about the diseases of the liver: infectious hepatitis, liver cirrhosis, cancer of the liver, gallstones. You can add yours, you can add others. Talking about the lungs, we have lung cancer, tuberculosis, pneumonia, bronchitis, asthma, common cold, whooping cough, severe acute respiratory syndrome, that's SARS, otherwise known as influenza. So these are about the diseases that affect specimen B and specimen C. Moving on to the next question, that's talking about question 15. We were told, what system does each of specimen B and C belong in animals? So talking about the liver, what system does it belong? We have various systems that makes organism a living entity, from the digestive system to the nervous system to the endocrine system, to the reproductive system, respiratory system, and what have you. So when you talk about the liver, you need to understand that the liver serves so many functions, but predominantly it falls into the digestive system. And when you talk about the lungs, that talks about the respiratory system. Moving on to the next question, talking about question 16. Of course, as you can see on your screen, question 16 says specimen B is a fresh liver lobe of a small mammal. Examine the specimen carefully. We were told to first of all describe the specimen. Of course, in describing the liver, specimen B is a dark red, soft but firm to touch. It is fairly large in size, and it has a cone or oblong, you call that oval shape, talking about the shape of the liver. So when we are told to describe a specimen, we describe based on what we see. It is dark red, it is soft when you touch it, it is firm as well, and it is fairly large in size. And of course, you talk about the shape, it is cone or oblong or oval shape. Looking at the next question, that's Roman figure 2. It says, mention the name of the structure you can see apart from the liver lobe and describe it. The other structure we can see aside the liver is the gallbladder. We were told to describe it. You need to know that the gallbladder is greenish yellow in color with an oblong or elongated sac-like structure, and when you touch it, it is soft to the touch. Once again, you can see the description. It's just about what you can see and what you can touch. Okay, that ends question 16, Roman figure 2. In Roman figure 3, part of the aside the specimen, there are certain materials that should be available in the course of this examination. One of them is a hand lens, or what we call magnifying lens, red litmus paper, blue litmus paper, office pen, tissue paper. So you look at the next question. It says, pierce the structure that's talking about the gallbladder with a clean, sharp, dissecting pin or office pin. Test the oozing substance, oozing content. That's the content coming out after you must have pierced with a pin with a red litmus paper. Of course, you know that the content of the gallbladder, talking about the bowel, is alkaline with pH from 7 to 8. So when you test with a red litmus here, as you can see, when you test with a red litmus here, the red litmus is going to turn blue. Yes, because why? The pH is above 7. The red litmus here is going to turn blue because the pH of the contents of the gallbladder, talking about the bile pigment, is actually alkaline with pH greater than 7. So testing with the red litmus here is going to mean that the red litmus paper is going to turn blue, and that ends Roman figure 3. Now, they said name the oozing substance and where it is manufactured. The oozing substance here is bile. The oozing substance here is bile, and it is manufactured in the liver. The oozing substance here is what? Bile, and it is manufactured in the liver. Then Roman figure 5, we were told to state 3 functions of the substance that we have named. So it's like asking us the functions of bile. That's the substance we have named. Of course, if you go back to previous question, we've talked about the functions of bile pigment before, so you can look that up. To Roman figure 6, we were told to list 3 functions of the liver and 3 diseases of the liver. So as you can see on your screen, talking about the functions of the liver, one of the very first thing is about there's something we call the liver insulin mechanism that regulates blood glucose or blood sugar. You can see it regulates blood sugar. It regulates the level of glucose in the blood. After a meal rich in glucose, that's a carbohydrate meal, you expect that the blood is going to be very rich in glucose. So when blood flows to the liver, what the liver does is that it removes excess glucose once insulin is released by the pancreas, converting excess glucose into glycogen for storage. Subsequently, when there is need, when the blood glucose level drops, the glycogen will be reconverted back into glucose. So what the liver does is that it maintains fairly constant blood sugar. That's talking about regulation of glucose. Also, I have said it before, talking about function number 2, the body has no protein store. So when we have excess protein in the blood, when I say excess protein here, the smallest unit, the monomer of protein, is amino acids. Acids we call it. So accumulation of excess acid in the body fluid, that's in the blood, is going to mean a lower pH. Amino acids, the pH falls. So imagine having excess amino acids just in the body fluid. That means there will be an alteration in the pH of the whole system, which can be very fatal. So what the liver does in this case is that when blood flows through the liver, excess amino acids are removed from the blood, and they are deaminated. They are deaminated. That's, in this case, they are converted into urea, some into uric acid, some into amino acid, depending on the organism. So we just basically say that the liver deaminates proteins. Also, the liver is responsible for the manufacture of bile, and this bile, is as a result of old and non-functional red blood cell being destroyed in the liver, and various chemicals, being destroyed in the liver. When you talk about the liver produces certain plasma protein like fibrinogen. Yes, it produces fibrinogen, things like globulin and the likes. And also the liver it produces it, which is distributed to all parts of the body. That's talking about distribution of it. There are so many functions. The liver stores iron resulting from the composition of hemoglobin. It converts other sugars to glucose and the like. So you can see all of that on your screen, talking about the functions of the liver. Also, we were told to list diseases of the liver. We've listed that before, so take note of that. And lastly, here we were told to suggest the system in the mammalian or human body associated with specimen B. That is the digestive system. We have talked about that before, and that just about ends question 16. So moving on to the next question, talking about question 17. This here talks about specimen D and specimen E. Of course, specimen D is a gizzard of a fowl. Specimen E is intestine of a fowl. So in question 17, we were told to give one characteristic or feature each of D and E that make them suitable for their function. So what makes the gizzard suitable for its function, and what makes the intestine of a fowl suitable for its function? Talking about the gizzard, as you can see on your screen, the gizzard is actually muscular. Why the gizzard is muscular is that what it does is to grind large food particles, breaking them down into smaller ones. From the gizzard, the smaller food particles then move into the intestine. And talking about the intestine is thin. And why is the intestine thin? So that when digestion is complete in the intestine, it is very easy for the digested food or the nutrients to be absorbed into the blood. So when you look at that question again, give one feature that makes specimen D and E suitable for their function. For specimen D, what makes it suitable for its function of grinding large food particles into smaller ones is that it is muscular. And what makes E able to perform its function of absorption of soluble food or nutrients into the blood is the fact that it is thin. Then for question 18, as you can see on your screen, it says we should give two functions each of specimen D and specimen E. So we should give two functions for specimen D, specimen E. What are the functions of the gizzard? It aids digestion by particle size reduction. I've said that there. It grinds large food particles into smaller ones, and also it regulates feed flow. That's regulation of feed flow. As you can see in that diagram, you can see the position of the gizzard. When birds, when they eat, the food gets to the gizzard first, where it will be broken down before it is slowly passed into the thin small intestine, from where digestion is then complete. So you see that gizzard sort of helps in regulation of feed flow, so that it is not too fast. And then also talking about specimen E, intestine of a fowl, the intestine proper, it is for digestion of food and absorption of nutrients or digested food. And with that, we've come to the end of the very first part of this biology practical, talking about probable question for specimen A to E. Quite lengthy, but it is what it is. It is biology practical, and it is usually like that. For myself and the entire team, it's bye for now.